What I'll do today is that I was supposed to teach the last couple of lectures, but I had to be suddenly I had to be traveling, so therefore Dr. Mishra taught those lectures. But before leaving, I had actually recorded one of those lectures, so I'll just play those. But there are some places in between where I need to intervene, so I'll intervene as and when required. And if you have any questions, you just type in the chat box because I'm here. So I will also answer those. And as I said, I will intervene as and when required. So am I audible? Can you all hear me? Could you see my slide when I changed the screen? Yes? Okay, great. So let's get started from here. All right. So as I told you last time, in today's class, we'll see how to construct another of the features that we have discussed so far, namely many body tensor representation. So you might recall that MBTR is one of the global features, just like Euler matrix and sign matrix. Just stopped it there. So I hope you are able to see and listen to my voice on that when I showed you the slide. I just need one confirmation. Okay, fine. So we will learn how to construct it. And before I go on to discuss how to construct many body tensor representation, I'd like to remind you what MBTR is, because there are various components to it. And so there are distinct ways to describe how to construct different different aspects or different components of MBTI. Okay, so what was the basic concept of MBTI? Um, you recall, so the core idea was that we identify various structural motifs. We identify structural motifs of different sizes, of 1 atom, 2 atom, 3 atom motifs. And we also consider the chemical identity of the elements that are forming that motif. And then we encode that motif in some way. And the particular thing was that the motif of, let's see if I can get, yeah. All right. So these motifs are captured by, so you capture the motif and then you map that motif to some scalar function g. K is the size of the motif, so k equal to 1 is just 1 atom motifs, that is individual atoms. K equal to 2 mean 2 atom motifs, and so on. And the R1 etc. up to Rk denotes the positions of the atoms forming that motif. And I showed you examples of this molecule HCN, which we worked with earlier. And here it's a general 3-body motif with atoms L, K, and N. So, okay, so how do you sort of construct or convert the motif, map the motif to a scalar function? If it is a one atom motif, then the scalar function is just the atomic number. And note that these G functions had this index Z, which is the atomic number. So there is one G function, one G1 function for each Z, each element that appears in your material. Or actually, when you are training using NVTR as a feature, there has to be one G1 for every element that appears in your data set. Okay, and that is G1Z is simply equal to Z. Okay, there's nothing much to do about it. But when we have two atom motifs, we take care of the distance. So that distance between the two atoms, and G2 for two atoms is simply the 1 over their distance. And so in this expression, we have not explicitly written the Z's, but it will of course depend on Z1, Z2. So for each unique Z1, Z2 pair, you will have a G2 function, which is just equal to the 1 over the pair distance. Similarly, the the three atom motif is mapped onto a function G3, which of course depends on the positions of the three atoms L, N, and M, and which was taken equal to the cosine of the angles between the vector between L and M atoms and M and N atoms. So in this case, M happens to be at the center. So this is a vector, that is another vector, and the angle between these vectors is taken as the G3 function. And once we construct these scalar functions, these are broadened. So this broadening was the next step using a Gaussian kernel. And I had shown you. So the philosophy was you identify motifs, map those onto scalar functions, broaden those scalar functions. And so given to broaden it, so G1 is how do you broaden it? So G1 this is the same way you can broaden it using a Gaussian kernel. Now, so now D1, the broadened version of G1, now becomes a function of some variable x. So when I say broaden, I mean, so otherwise you see G1s were just discrete functions. It is equal to Z1 when Ri happens to be the position of the atom Z, atom with atomic number Zi. But now we are sort of broadening it into a function D1, which is a function of a continuous variable x. And so the question is, what is the range of x that we should choose? Of course, that depends on what is the range of values that your G1 function will take, can take in your data set. Right? So for example, if we have, say we have materials which are made of atoms, let's say if I, just think of the periodic table, the elements that appear in these materials range from Z equal to, let's say 1, which is hydrogen, say up to, it could be whatever, but suppose it is 17, which is chlorine. Okay, so as far as G1 is concerned, we will never have values which are beyond 17, but then we are broadening it. So a sharp peak at Z1 equal to 17 will get broadened into a Gaussian centered around 17. So we may want to go a little beyond 17. How much? That depends on the parameter sigma 1. You see, sigma 1 in the Gaussian determines its width. So depending on sigma 1, you may want to go a little beyond 17, and similarly, you would like to go a little beyond 1. So maybe you can start from 0 and then go somewhat upward of 17, maybe 18 or 19. I'm just throwing numbers at it. Of course, it will depend on the actual value of sigma. I'll show you an example of the sodium chloride solid. Sodium has atomic number 11. Fluorine has atomic number 17. So the G1 function will, before broadening, will have peaks only at 11 and 17, but then we'll broaden it and we'll see the plot. Fine. So that is G1. Similarly, broaden G2 into a function D2, which is a function of x. And what is this x? It's an inverse distance. So you have to have some idea about the interatomic distances, and depending on that, inverse of that will set the range of x that you can have in your system. All right, and again, sigma 2 in this case will determine how much is the width. So you will have different pair distances, and each pair distance will give you an inverse value. So you will have peaks around those inverse distance values, and then sigma 2 will broaden those peaks. How do you choose sigma 1 and sigma 2? These are, the best way of choosing these is to essentially try different values of these parameters, and then whichever gives you the best result, you choose. So you have to do some tests, see which one gives you, maybe with a smaller training set or whatever, computationally feasible, and then when you do the big calculation with all your data, you use a set of values. Okay, so these are D1, D2. Similarly, you can broaden D3, and D3 obviously it's a cos function, so therefore it can only go from -1 to +1. So the range is obvious, and you can set it accordingly. And then once we have calculated, say, the D1s for all the elements, then there are different elements, the same element occurring at different positions. We calculate the G1 for all of them, for each one of these Ri's, and then we sum them over, and we discuss these weights. So as far as the G1 calculation is concerned, or combining D1s are concerned, the weights are all kept as 1. So that gives me one of the MBTR with K equal to 1, the one body motif, and that is called F1, and which is a sum over all these D functions. Similarly, the two body MBTR, F2 for different Z1, Z2 would be sum over the D2s, but weighted now. So how do we choose the weights? And we argue that one reasonable way to choose the weights is so that they go down exponentially as the distance between the atoms Ri and Rj increase. The same way, the three-body MBTR is constructed. And I had shown you the weights. So the weights are all 1 for one body motif. Weights are all 2 for weights are exponentially damped for 2-body and 3-body MBTRs. Yes. And we discussed how the MBTR for a water molecule is constructed. I mean, we did not discuss how to construct it, but basically how to make sense of these plots. All right, so we had discussed these things. So let us now see if we can all right, so what I would like to show you is how to construct MBTR. So we will learn how to construct MBTR in general, and I will illustrate it by constructing MBTR for a sodium chloride solid. Sodium chloride has an FCC structure, basically two interpenetrating FCC lattices, one of the sodium atoms, one of the chlorine atoms. So if you look at this figure, carefully, long enough, you will easily figure out that the green spheres, which are the chlorine atoms, form an FCC lattice, and similarly the purple ones, which are the sodium atoms, also form another FCC lattice, but they are displaced with respect to each other. So I'm not worrying about the further details of this crystal structure at the moment. What is the lattice constant? What is the sodium chlorine distance, etc., etc. You can open any textbook on basic solid state physics and you can find this out. Okay, so all I'm going to discuss is how to construct MBTR for this. So I'll open, I'll try to share my terminal and show you how to do it. Yeah, are there any questions? These are things we had discussed earlier, so I believe it is clear enough. And I also wanted to ensure that there are no difficulties, technical, in viewing this. Okay, I don't see anything. If there are no questions, we'll just continue. All right. So let me try to, I hope you can see it. I'm sharing my terminal. So this is a script which constructs MBTR if you give a structure and in the form of a C file. So we have seen how structures are stored in JSON files, but we have not, at least I have not shown you C files, but it's a very standard format, crystallographic information format. And just as AAC can reading structure information from JSON files, AAC can also reading structure information from C files. And many databases, for example, even when you access data from materials project, I showed you that we were storing it as JSON files. You can also decide to store it in C format. If you download data from, say, the ICSD database, you will get structures in C format. Okay, so these are usual things that you have to import. Okay, you import atoms from AAC, AAC input output read, formula we really don't use, AAC database connect also, in this case we'll not use it, but some of the headers I just happen to have files. Okay, the important thing is here. From these scribe descriptors, you import MBTR. So what is that library? What is that function that you have to import from these scribe descriptors? You can check the GitHub page of Describe, go to their documentation and check it. So when you are constructing MBTR, you have to import it. This is the line that reads in the structure information from the C file. Okay, so I have in my area a file with this file name, and I'll just go out of this file and open that just to show how a C file looks like. And this is something I have downloaded from the ICSD. So this is sodium chloride. As you can see, it contains sodium and chlorine. One chlorine, one sodium, density, etc., etc. You know, So it will give you some information about crystal structures. You don't have to understand all of it, but these are, some headers. So journal. So it will give you the full journal information where this structure was published. So here are the author names that you have. Okay, and here is the journal reference. And then you have the author names, and then cell length. So the 3 cell length, and because this is a cubic system, all lattice constants have equal length, 5.63. Then because it's a cubic system, all lattice angles are 90 degrees. It will give you the cell volume. Okay, there are 4 atoms. Unit cell. These are the last line in the highlighted portion. Tell you what the space group of the structure is. There are different sort of notations that are used to indicate space groups. So it tells you in 2 different ways. Third, this line tells you the space group in another way, which is given by the number. So there are various ways that same space group can be specified. So it gives you all of those. And after that, this part essentially somehow captures or contains information about where the atoms are located in the unit cell. So not worry about all the information that it contains. But basically that's how a file looks like. You don't have to sort of understand it line by line, but you just have to know that given a proper C file, AC read can directly read the structure from that C file, which is done in this line. Then for MBTR, so there are 3 components of MBTR. MBTR for K equal to 1, that is 1 atom motif. K equal to 2, 2 atom motif. K equal to 3, 3 atom motif. And that's how we discussed. I'll show you two of these. So for K equal to 1, K equal to 2. So first I will comment out the sections for, or first I can show you for K equal to 2, and then I'll get back to K equal to 1. So this part sets the calculation for K equal to 2 MBTR. All right. So what do you have to specify? Species, that is, what are the different types of elements that you have in your materials? The same thing as we saw in case of sign matrix and ACSA. Then the next keyword is geometry, and function is inverse distance. So that is exactly what I told you, that G2s are taken as inverse distances. However, so this was the initial version of these guys. Later on, I think they changed it, and there are other ways you can capture these interatomic distances for 2-body MBTR. So if I can, I will try to just let me see if I can go to that. So I have to stop sharing this. And I would have to share something else if I can. So let me get here. So now I'm sharing my browser. So we are talking this K equal to 2 MBTR. So inverse distance is one way of capturing information. You can also simply say distance. So in that case, G2 would not be 1 over the distance Rij. It would be just the distance Rij. It's just fine, except that the x, the continuous variable, when you broaden it into the D2 function, would appropriately change. So this is a later addition in this, right? All right. And note that although we are talking of k equal to 1, k equal to 2, k equal to 3, you never set these explicitly, you never set k explicitly, okay, in your script. So I'll stop sharing this. I'll go back to sharing my terminal. Yeah. So the fact that you are talking about or you are trying to construct two-body MBTR is understood by describe from the fact that your geometry is either distance or inverse distance. All right. So it will understand that you are talking of two-body MBTR. Grid is this is the minimum value. So this continuous variable x, what is its minimum value? What is its maximum value? You can set those. Sigma is that sigma 2 parameter, the sigma of the Gaussian broadening. And n is basically the number of points. So x is a continuous variable, but when you are calculating it on a computer, you have to calculate it as at some discrete number of points. You typically would want to calculate at many more than 25 points, but because, I mean, just to keep, so that I can do the calculation and show you, I have kept n equal to 25 here. And these weights are just the weights I talked about when you combine D2s linearly to construct the F2 function. So in this case, we use exponential, and there is a scale factor. There is an s in the exponential also. So it is e to the minus s times the distance between the two atoms. So it is, the scale is that s. And threshold means if the weight falls below this number, then you don't consider those pairs. If they're too far apart, so that the scale, so that this exponential function is less than 10 to the minus 3, then we don't consider their contributions. And we are periodic function. We normalize a particular MBTR, etc. So you can check the describe documentation to understand further details of it. And so this weightage is what function you are using, and probably they now have more options than exponential, so you can check it, but we're just going to do it with exponential. So this steps as to how this is to be calculated, and this line then calculates MBTR for the current MBTR2 for the current structure. All right. So the idea is the same. In one statement, you tell describe what are the parameters according to which a particular feature has to be calculated, and then after that, in another statement, you tell you pass on the current structure so that describe can actually calculate MBTR. So we will try to calculate MBTR. That file, I hope it works. Yeah, so that's the MBTR. So that's what we have calculated, and note that I have, so it says 75 grid points. Okay, species are 11 and 17, sodium and chlorine, and there are 75 grid points. Why is it 75? Because we specified 25. Note that there are sodium and chlorine, so there are sodium-sodium pairs, there are chlorine-chlorine pairs, then there are sodium-chlorine pairs. 3 different pairs of atoms. 3 times 25 is 75. So now what we do is we just run the same thing, but we save it to a file. And then I have a separate file. You can do it in the same Python script if the versions of Python match, but because these for describe, every time I have to fall back onto sort of lower version of Python, certain other things don't run, so I have to take this two-step approach. So this is a simple Python script. I don't even need to show you, which reads in that temporary file and plots the MBTR for us. So I'll show you. It looks, yeah. See, there are 3 peaks, and whether the locations of the peaks are correct or not, in order to understand that, you have to work out the chlorine, sodium, chlorine distances. You have to look at their inverse and see everything is working out fine. I haven't done that, but I'm just showing you that you. Okay, so I need to come in here, and I would like to show you that. Yeah. So here is the plot that that is not there in that video. So the x-axis is related to the inverse of the distance. It's that scale, but don't read these numbers. These numbers are just the sort of serial number of points along that axis. These are not to scale, so don't try to read those. But you see there are 3 peaks which correspond to basically 3 pair distances that has been captured by this plot. This is MBTR2. I'll show you another example later on where MBTR2 has been calculated with G2 as the distance rather than the inverse distance. Okay, so let's continue. Just showing you that you get 3 peaks as you expect for this material, for 3 pairs of atoms. This is the same kind of information that was plotted in that paper that I showed you for H2O, but our curve is not very smooth because the number of 3 points we have is very small. All right, so that is MBTR2. We will now go back to describe, and I'll show you how MBTR1 works. So I'll comment this out. I would like to calculate MBTR1. I would comment this, and I would uncomment these. Okay, so this is MBTR1. What are the things here? Species with species, again the same thing. What are the elements that appear in the data set? Geometry function is just the atomic number, and that's the only option you have for G1. G1 R1 Ri is equal to Zi. And because my system contains sodium and chlorine with Z equal to 11 and 17, I have set the range of this function from 8 to 21. And I calculate it at 50 different points with a sigma of 0.1. I mean, these are not something I have chosen carefully. This is just a demonstration. Okay, if you're willing, if you're trying to do serious calculations, you have to choose these quantities carefully. Weight function is unity. Again, I think that's the only option for one body in MBTR. So that is it. I will then run it once again. Okay, I was trying to print something which is not calculated.
this case. MBTR1. Yeah. So now I'm printing the length of MBTR1 and printing MBTR1. So let's see if it works. Yeah. So there are 100 grid points, and along those 100 grid points, which are just the Z numbers between 8 and 21, the limits I chose, it is calculating the F1 function. So again, I'll try to plot it. I store it to 10.5, and then I have another plotting script. I just run it, and here is the plot that you see. So you see, yeah, so I have to show you the plot, which is not there. So here is MbTr1 for sodium chloride. We have two elements at 11 and 17, Z equal to 11 and 17. We don't have the peaks exactly at 17 and 11, but probably one has to choose those parameters carefully. I didn't do enough testing with the parameters, but that is something I leave for you as a task. You can do it, but we do get two peaks that we would expect. So that is the point. One peak is around 8. Not exactly, sorry. It has appeared somewhere below 10. Maybe I have to choose my parameters carefully. And this has appeared somewhere above 18, although I was expecting it around 17. But that is where I have to choose my parameters carefully. But you see two peaks. Okay, so that is the point. So now, if you want to use MBTR as your descriptor or feature for any machine learning task, train your model, what you have to do is you have to concatenate these components for k equal to 1, k equal to 2. You have to sort of concatenate all of them so that you get a total MBTR, which contains all one body, two body, three body, if you want, interactions, motifs, structural motifs. So I'm not going to show you three body terms, but again, if not sure my screen sharing worked properly. Okay, so I can skip some part of it, and what I'll do is I'll show you a plot for, let's see, I'll show you a plot of MBTR2 with G2 being the distance between the pairs of atoms. So instead of being inverse distance, it is taken as the distance. And I use a different sigma for this plot. And this is something you can work out. If you take the sodium chloride structure and you ask what is the nearest neighbor distance between any two atoms, you will find that the nearest neighbors are sodium and chlorine with a distance which is roughly equal to 2.8 angstrom. And if you look at this plot, the first peak appears at 2.8. Then the second peak would be between two chlorine or two sodium atoms, I believe. And that distance happens to be around 3.9, if I'm correct. It's somewhere between
somewhere around between 3 and 4. So I see a small peak here. Then there is a peak just above 3.5. So the locations are not exact from this, but again, this is calculated. Then several quantities are summed over, so that produces this entire plot. So you don't try to match exactly, but at least you can see that the first peak appears somewhere around 2.8, and then there are peaks around interatomic distances that you can calculate. So you can calculate first neighbor, second neighbor, third neighbor distances from the NaCl structure and see how these match. So this is an exercise. You can also, so this will be an exercise for you to calculate MBTR and then match with the distances you can calculate by hand. Okay, so that is it. I'll try to put these in my slide, but you should calculate these things. Let me check if there are questions. No. Yeah. Am I audible, visible? Is my video visible? Yeah, seems like. Yeah, any questions? Okay, let's continue. I'll stop sharing for a moment. So we learned how to calculate different components of MBTR. And then if you want to use MBTR for model training, for prediction of some properties, you have to combine all these components. So you concatenate them basically and use that as a feature. So we have talked about various features. I showed you examples. Now what I'll show you is that one application, it is discussed in the archive paper for the described library that I have shared reference with you on my slides. I have taken some results from that paper, and that is what I'll show you, how various features that we have discussed so far perform when you try to predict formation energy of materials. So again, I have to share my slides. Yeah. So next slide, this one. So performance of various features for formation energy predictions. Data has been taken. It's the same paper that introduced these, right? Data from OQMD database, you will have linked to OQMD on my slides. And what they have chosen are materials that have maximum of 10 atoms in unit cell and a maximum of 6 different elements in the unit cell. And that way they found 222,000 materials after some not properly converged structures were removed. The model they used was KRR, that is kernel regeneration model, trained to predict heat of formation or formation energy for different feature vectors. So the training data set is same, the features are same, features are changing, but different sets of features. So these are different exercises, and then they are performing how a model is the same. So the data set is the same, model is the same. What is changing are the features, and so that way you can sort of test how different features are working. So different features. So there are two plots you can see. On the I'm talking of the left plot.
And what is plotted along the horizontal axis are the training set size. The different number of samples used to train the models. And as expected in any machine learning model, as the training set size increases, model performance increases because it is trained better, it is learning better in underlying function. So apart from that, so I will not worry about the training set size. That variation is kind of trivial, or I wouldn't say trivial, it's kind of expected. So we'll focus on these points at the extreme right on this plot, and different colors or different types of lines represent different features. And see, the best performance is obtained for SOAP, the smooth overlap of atomic positions with Gaussian type orbitals. Okay? Then the next higher point. So what is plotted along this axis, the vertical axis, is the mean absolute error in the prediction in EV per atom. So the smaller the mean absolute error, the better the model performs. So best performance is obtained using SOAP. The next one is MBTR, combining k equal to 1, 2, and 3 MBTR components. Okay, so all three included. The next one, I have to look at it closely. This point corresponds to sign matrix. This point, the light green one, corresponds to Coulomb matrix. And this one at the top, the highest one, which is the worst performing model, is with Ewald sum. Now here is a very strange thing, you see. Coulomb matrix should ideally make no sense for a periodic system, right? Because they have just calculated the Coulomb matrix for the atoms in the unit cell, but that does not take into account the periodic nature of the crystal. Yet that Coulomb matrix as a feature is performing better than the Ewald sum matrix, which takes into account the full periodicity of the structure and does calculation of a proper calculation of a Coulomb interaction between pairs of atoms. Why? No understanding. It's just that for this quantity, and the model is not really learning the physics. That is something you must understand. The model is learning some underlying pattern. It's not learning causality. It's learning some correlation, some pattern. And as far as that is concerned, it does well with the Coulomb matrix. So we should not try to dig deeper into this and understand what is going on. And that, at least that is my sort of position. On the right is something very interesting. Again, training set size along the horizontal axis and mean absolute error along the vertical axis. And here different lines are for different components of MBTR. So the lowest one is MBTR when all k equal to 1, 2, and 3 MBTR components are taken. So that gives the best performance. The next higher one is for k equal to 2. Okay, so the top three here are individual. So individual MBTR components have been taken as features. So right. If you take individual features, A2 seems to perform the best, followed by 3, and the MBTR1 performs the worst. Not surprising. You are only taking information about the constituent elements, their Z values, and nothing. Okay, so that sort of goes with our physical intuition. So this is just an example as to how different features perform in prediction of formation energy for inorganic materials obtained from OQMD. Will the same trend hold when you take, say, bigger data sets, combine different databases to come up with a bigger database? I do not know. And what about the absolute numbers of the mean absolute error? So here you see even in the best case, the mean absolute error seems to be of the order of 100 MeV per atom. There is 0.1 EV per atom. So there are now models, or there are other representations which now can produce mean absolute errors that are much smaller than this. But then again, in those cases, perhaps the training sets are also much larger. So it always becomes very hard to compare models unless you change only one thing and everything else is kept constant. So there are features, there is a data set, even for a given model. So you can only change one at a time to understand how which one is working better or which one is working worse. All right, so that's all. I will share the script and the slides. It might take me a couple of days, but I will eventually share these. And I would again strongly recommend that you work with the scripts, you try to do small examples, and only that way you learn these things. The problem with an online course is that we cannot.
Yeah, so I can stop that and I can come in here and repeat what I wanted to convey is that the course, when we designed it, we wanted to have a significant portion of hands-on session. But then it becomes very difficult in an online mode to really see what you are doing, if you are having any problems, to sort those out, etc. But as we had declared earlier, we will have a few sessions, a few classes in which we will discuss these things with you. You are, you should come with your laptop or computer or whatever you are working with, and we would like to sort of interact with you, so you won't be this is particularly true and mandatory for those who are doing the course for credit. So Monday's class will be an interactive class like a tutorial. You are asked to come with your computer. We will ask you, and note that for you, those of you who are crediting the course, attending that class will be mandatory. We'll have one more session or maybe more sessions later on. um, so essentially we will have an interactive class, and also you are free to ask questions or point out any technical difficulties you might be having. Okay, so that is what I wanted to convey today. I'm not sure if Shubhankar, are you there? Not sure he's there today. Um, anyway, so we will meet on Monday. If you have any question on today's class, you are free to ask or type in. I'll wait for a couple of minutes and then we will close. Okay, so to repeat, Monday's class will be an interactive class, and those of you who are crediting the course, it is mandatory that you be present. Okay, so I'll stop here.